Shoji is the professor in University of Tokyo and an expert of hybrid systems. So he has been working on a biohybrid system, and he has been working on many different exciting projects about how the animals' bodies, biological bodies, and machine can come together. And this is actually one of the really central topic of embodied intelligence: what makes distinction between biological systems and the machines, and how we can put them together in the future so that we can actually make a more feasible biologically feasible embodiment in the future. So I'm very excited to have you here in this Embodied conference as a plenary speaker. I think I can give the floor to you, Shoji. Can you just turn on your microphone and start your presentation? Thank you very much.
Yes, thank you very much for introductions. Oh, can you hear me?
Yes, we can hear you very well. And please go ahead.
Okay, thank you very much, Professor Ida, for the very kind invitations to the very, I'll say, fantastic conference like Embodied Intelligence. And today, I'm Shoji Takeuchi from the University of Tokyo. And today I'm going to talk about the, let's say, biohybrid robot. And what is a biohybrid robot? The biohybrid robot is the kind of the consists of the two kind of things. One is the biological materials. The other one is the artificial systems. And this is one of the cartoon, I would say, expressing one of our goal of the biohybrid robot, which consists of the basically consists of the how to say humanoid structures, but covered with a living skin. So that they consist of the living cells. So why living cells is important? Because the cells have the very nice sensors that can react to the single molecules. So that's the robot itself has the highly sensitive and selective sensors. And also cells has a, I would say, the biological function like self-healing or self-reproduction ability. So that once it's, how to say, make a damage, they have the self-healing ability of the robot. And also this robot actually actuate by the power of the living muscles. So the muscle advantage of the muscle is the highly energy efficient, and they can achieve the soft and flexible motions. And actually they actuate without any noise. So the silent actuation is possible. And once we can make the, how to say, muscle actuator coming from the cells coming from the cow or beef, so that we can actually eat it after using the robot, because the muscle is the kind of the muscle becomes a steak or other type of the meat. And also to keep the cells alive for long times, so we need to, how to say, we need to create the culture media. So those kind of culture media can be produced by the kind of the organs, like livers or, how to say, intestine and so on. So those kind of the organs can be made by the human iPS cell derived cells and so on. So that the tissue engineering approach is also very useful for making a biohybrid robot. Okay, so this is again, this is a kind of the one of the goal of our biohybrid robot. So let's say some examples. So okay, so why we need such kind of biohybrid robot? So because the engineer creates the various type of the machine, like a humanoid, smartphone, or self-driving cars. So that can actually, I would say, change our world much better and convenient. But yet we cannot create the, how to say, very attractive functions that the biological system have in natures. For example, we cannot create the very highly sensitive sensors like dog nose, or how to say, the cells has a self-healing and self-reproducing ability. And also the muscles and brain is very highly energy efficient systems. Okay, so that if we cannot create whole biological system from scratch, why not why not using the biological systems and combine with the artificial systems? That is our concept of the biohybrid systems that we are now working on. Okay, so I will give you some example of our biohybrid systems. Of course, we want to make these kind of the robots in near the future, which is a kind of humanoid covered with a living skin. But since if we want to make a really large humanoids, we need a bunch of their cells. So that the first trial to make this kind of robot, we actually focus on making just a finger. So this is just a one finger of the I would say humanoid, but having the living skin over the I would say finger-like robot, so that it looks like a I would say human skin. So how we can make it? So Mr. Kawai, who is a Masako student of my group, he actually made a really nice job. And at first, he made the 3D printed finger bone-like structures like this, which has a 1, 2, 3 joint like this. And after making the 3D printing structures, we actually put the finger onto the small chambers and put the collagens with some dermal cells. And after culturing the 2 or 3 days in the small chambers, the cells can, I would say, contract the collagens, and the collagen volume gets to shrink and shrink. And it covers the surface of the 3D printed finger bone-like structure like this. And after these structures, after making these structures, we actually put the another type of cells, that is a kind of epidermal cells, onto the dermal structures, so that we can basically make the dermal structure, dermal tissues with an epidermal skin. So those kind of the structure is very important to mimic the human skin. And after making the after culturing the epidermal tissue, epidermal cells onto the finger bone-like structures, so we can actually make the finger finger robot covered with a living skin like this. And those kind of the robot can actually actuate by the power of the motors. And the surface, I mean, the texture of the living skin looks like a human being. So we can actually collapse the epidermal layer, and the epidermal layer actually, I'll say, repel the drop, so that they have their barrier functions. And what good things of the living skin as a part of the robot, so we can actually put the biological function like a self-healing ability of the robot. For example, if we make a damage of the tissues of the finger bone, like finger robot, and once we cover the collagen sheet onto the damaged area, so that we can actually heal the damaged area after, I'll say, 1 week cultures. So because the near the damaged area, so we still have the living cells like this, and the living cells want to proliferate and migrate into the collagen area, so that we can actually cover the damaged area within the 1 week cultivations. So that's those kind of the materials very much useful for the robot, I believe. Okay. So thinking about the biohybrid machines or biohybrid robots, so we can actually divide the 4 type of device as a biohybrid systems, which is a kind of the sensors, reactors, and actuators, and processors. So sensor is a very understandable. So if we can make, if we can use the biological materials as a sensor part of the systems, so we can actually make the highly sensitive and selective sensors compared to the artificial sensors. And the biohybrid reactor can mimic the biological reaction in our body. So if we can make the, I'll say, drugs or medicines, so we can normally, so we actually, how to say, investigate the effect of the drugs by using the animal testing, by using the rat or how to say, monkey and pig and so on. But those kind of things is a kind of the ethical issues and far from the human beings and so on. so that if he can actually make the human, human organs within the small chips, so we don't have to do the animal testing and so on. So those kind of the reactors might be very useful for the pharmaceutical applications. And also, if we can make, if we can actually mimic the biological reactions in our body, so those kind of the chips is also work as an artificial organ in our body, so that these kind of systems is also work as a cell therapy applications and so on. Okay, so the biohybrid actuator I will mention right after this slide, but I'll say if we can use the living muscles as the actuator of the robots, so we can actually generate the highly efficient motions or silent motions compared to the artificial actuators and so on. And a biohybrid processor is a kind of far future technologies. That's the thinking about the neural engineering research field. They actually culture the living neurons on the chip and make an artificial network and get action potential from the each cells. So in near the future, if we can control the network by using the living neurons, so we can actually make the artificial living neurons for the some calculations or learning and so on. So those kind of the chip might be very useful for the highly efficient calculation or processing. Okay. So actually, we are now working on the 4 different kind of the biohybrid devices, including the sensors, reactors, and actuators and processors. So for the sake of times, I just want to explain about the story of the biohybrid actuators and the biohybrid sensors. Okay, the first, the biohybrid actuators. So in 2018, we published the Science Robotics papers, which is the kind of first paper coming out from a group like a biohybrid actuator can be used for the robotic applications. So this is a kind of finger bone-like structure. Biohybrid fingers consist of the finger bone, 3D printed finger bone-like structures, and the tissue engineered living muscles. We actually culture the skeletal muscle cells into the skeletal muscle tissues and combine with the finger bone-like structures. And once we apply the electric voltage between the living muscles, so they make a contraction motions and then pull the fingers back and forth like this. So using this kind of technologies, we can actually generate several kind of the biohybrid actuators like this. And those kind of the finger can pull up the small object. So this demonstration is very simple, but I think the world first demonstration where we can actually use the living muscles for the robot applications. So how we can make such kind of actuators? Dr. Yuya Morimoto, who is now a professor of the Waseda University, and he actually develops a very nice method to make a very thick muscle actuators. So we actually use the collagen hydrogels and puts the skeletal muscle cells dispersed into the collagen hydrogels. And using the PDMS stumps, so we can actually make the collagen structures like a sheet, cell-embedded collagen structures like a sheet, like this. And that kind of sheet has a small slit in the lateral directions. This structure is very important to keep the muscles fibers aligned to the single directions. Okay, so because they are in collagens, the skeletal muscle can grow and migrate inside the collagen area. And once the cells attach each other, they actually fuse. And then makes the muscle fibers like this. So this is a kind of the differentiation of the muscles. So during the muscle fiber formations, so thanks to the slit like this, the cell fiber can align onto the, only to the single directions, like a lateral direction. They cannot make the this directions, but they cannot make the muscle fiber at this directions. Okay, so that using this kind of sheet, So we can make the biased, multiple sheet and stack the sheet on top of each other so that we can make a very thick tissue within the short time. And after the cultivation of the sheets, so cell can grow and migrate and make fuse each other and make a muscle fibers. And again, the muscle fibers align to the only on to the single directions. So this morphology is very important to for the muscle tissues actuate to the single directions, because after applying the electric voltage between the muscles, they make a single contraction like this. So this is actually a photo how the muscle tissue looks like. The scale bar is 2 millimeter, so that the length of the tissue is about the let's say like a 1 centimeter in length and so on. And again, if you apply the voltage between the muscles, they make a contraction motions like this, one by one. Okay. So using these technologies, so one of my students tried to make a bio after publishing our work. Okay, so using these technologies, so we not only apply the robotics, robot application, but also this technology might be useful for the food applications as well. Because if we can make the actuator by using the cells from the cow, so we can actually make a beef steak without killing the animals. So this is very good for not only for the animals, but also the environment as well. So using this technology, we make the centimeter scale steak. So this is a kind of the world's first beef steak, which has the length of the 1 centimeters, and the thickness is about the 8 millimeter and so on. So anyway, very I would say very thick. And this is a kind of the actual movie how it looks like. So using the chopsticks, so it's very soft, looks like a raw meat. But the scale is very small, unfortunately. So we are now scaling up the structures, not only from the 1 centimeter, but also the 7 centimeter by 7 centimeter, and the thickness should be 2 centimeters, which corresponding to the 100 gram of the meat in the other features. And this is kind of the Japanese first tasting ceremony of the eating the cultured meat. So unfortunately, we cannot eat the very thick ones, but the thickness is about, in this times, the thickness is about 2 millimeter, which looks like a kind of shabu-shabu meat in Japanese. And but it looks like a real meat and having a lot of cells coming from the cow. And I am the kind of first guy in Japan who eats the cultured meat, cultured beef like this. And taste was not so, bad. So it tastes like I can taste the salty and the umami taste. But unfortunately, I cannot taste the taste of the beef. It's far from the taste of the beef. But that is a kind of the very surprising things, because the maybe over the 70 or 80% of the these tissues coming out coming from the beef cells, cells coming from the cow. But once we make the tissues by using the cells from the cow, so we cannot taste any taste of the beef. So that we are now focusing on at what stage or what conditions is very important to make the beef steak. That's kind of that's actually shows the taste like a beef. So that is kind of the basic research that we are now working on. Okay. Okay, that is a kind of story of the our biohybrid actuators. Okay, so the next is a story of the biohybrid sensors. So if you think of the biosensors has a long history over 60 or 70 years. But principle of the biosensor is very simple to detect the biomolecules. And if you have the biomolecules, so we need to design the receptors. That kind of receptor can be the DNA or protein or nanomaterials. that can bind the biomolecules very specifically. So that the selectivity should be very high. But the problem is, and the principle is very simple. And when the biomolecules bind to the receptors, they actually make some signals like electrons or photons or change of mass and so on, so that we can actually detect the presence of the biomolecules by measuring the presence of the electrons or photons or change of mass and so on. So that's the that is kind of the simple principle of the biosensors. But the problem of the current biosensors is when the biomolecule bind to the receptors, they only generate the single electrons or single or two photons or something like that. Only a few photons coming out from the receptors. And also the change of mass is only a single molecule, so that the mass change is very, very small. So the problem is how we can increase such kind of signals to detect the single molecules. That is a current problem of the biosensors. But if you take a look at nature's, so let's say our living cells, they actually have the super sensors to detect the single molecules, which cause their membrane proteins. And if you see the membrane proteins, that exist on the surface of the cells. And if you take a look at the surface of the cells, so we have the living membrane, lipid membranes. The size, thickness is about 5 nanometers. And in the membranes, they have the several membrane proteins that works as a sensors or gateway of the cells. And this kind of sensors, this kind of the membrane protein works as super sensors in the living cells. So this cartoon shows how it works. And here is the cell membrane. And if you have the single membrane proteins like this, they can actually recognize only a single specific molecules out of the bunch type of the molecules. Okay, so even if a lot of the molecules outside of the cells, they can recognize only a single molecules attached to the membrane proteins. So the selectivity is really high, like protein enzyme reaction and so on. Anyway, so after binding the single molecules, they actually change its conformation from the closed state to open state, like this. And after opening the membrane proteins, since there is some potential difference between the inside and outside of the cells, so there are a lot of the ions go through the membrane like this. So those kind of ionic flow can be measured by the special measurement machines. And the current generated by the ionic flow is on the order of picoampere. So picoampere is very small, but we can actually measure by using the preamplifier and so on. So if you think about the picoampere, so it's kind of the 10 to the 7 molecules per second. So that means these sensors can generate the single molecule signals to the 10 to the power of 7 molecules. So those kind of the amplifier cannot be made by using the artificial sensors so far. So that we think the membrane protein is kind of the super sensors that exist in our body. So that we want to use this kind of power into the artificial systems. So if you think about the membrane proteins, so we can actually focus on the mammalian systems or insect systems. But according to the mammalian systems like a dog nose, they have a lot of the molecules is needed to detect the single molecules. For example, once the molecules bind to the one membrane protein, they make a several cascade, and after that, the ion channels can open. So those kind of systems can be made maybe, but it's very difficult to reconstruct so far. So that's but on the other hand, if you think about the system of the insect, so the insect has a very simple systems, like they actually detect the molecules by using only ion channels. So if you have the ion channels, and if the ion channel detects only a single molecule, they make a ionic flow like this. So that's why we first use this kind of systems for detecting some molecules. So because the insect can also detect a very nice, I'll say, molecule, detects a very nice molecules in our societies. For example, bee can detect addictive drugs or bombs, and a mosquito, as you may know, can detect human beings and attach to the human skin and get the blood and so on. So that the first we try to use the insect senses and expressing the artificial living cells, and put into the microfluidic devices and insert the electrodes like this. And once the molecules, odorant molecules come from the inlet of the microfluidic channels and they attach to the membrane proteins, they make electric signals and like this. So once the molecules coming from here, and they can actually generate the electric signals, and the electric signals goes to the amplifier and then goes to the motor so that the robot can move. Okay. So this is a very simple demonstrations of the biohybrid sensors in the robot. So to detect the molecules, so we actually use the two kind of molecules. So one is a pheromone A, the other one is a pheromone B, because we actually use the insect membrane proteins. And but interestingly, so the size of the molecules and the structure of the molecules is very similar. And the weight, molecular weight is also very similar. So if the weight and the shape is very similar, so we cannot basically distinguish two kind of molecules, even if it's a very expensive, analytical machine. But the living cells or membrane protein can easily detect the difference between the two kind of molecules. For example, if he puts the pheromone A, nothing happens, so robot doesn't move. But if he puts a pheromone B, so we can actually see the signal difference in the generated by the membrane proteins, so that the robot can actually moves back and forth. So that's the biohybrid sensors works very well to achieve the highly sensitive and highly selective detections. And those kind of sensors, I think we can actually apply the various type of research area, like a blood testing, because the blood has a lot of the molecules, and if you can get the only single molecules which cause some disease, so we can actually detect the disease by only a single drop of the blood. And also we can detect some odorant molecule coming from the food, so that we can actually identify the freshness or product of the place and so on within a very short time by using the very handy sensors. And third application is we are now working on, which is the kind of the human detections in natural disasters. Because in Japan, we often have the natural disasters like a tsunami, earthquake, and typhoon, and so on. So, the finding the human being in disaster as soon as possible is very, very challenging issues. So currently, the dog works very well. They can find the human being in a very short time, like within the 5 minutes and 10 minutes and so on. But the concentration time of the dog is only 15 minutes or less. So that's they cannot work for 24 hours. So, but if you can make the nose sensors or molecular sensor like dog nose and put onto the robot can work 24 hours in finding human beings. So that might be useful compared to the living dogs and so on. And to do so, we also focus on using the sensors coming from the mosquito, and because the mosquito can easily find the human beings. Okay, and we actually identify the DNA of the single membrane protein that can react to the sweat of the human. And we can actually generate the membrane protein a lot by using the cell-free systems and reconstruct to the cell membrane like this. And in this census, we actually form the artificial lipid bilayer, which is a kind of the artificial cell membrane, and which has the membrane protein sensors of the mosquitoes. And once the human odors attach to the membrane proteins, they actually change its form from the closed state to the open state, and it makes a ion channel, ion current through the membrane. So that once we can measure the ionic flow by using the sensors, so we can actually detect the human odors very easily. And that kind of sensor signals go to the also motor by using the wireless transmissions. So the motor can move. That means the robots can move. Okay, so this is a first results coming out from the robot. So that we can, when we put the membrane proteins and the, okay, so if you don't put any, sweat of humans, odorant molecules coming from the humans, so that we don't have any signals like this. But if you put the human odors, like odorant molecules coming from the human, and then we actually see the open close signals coming from the ion channels, because like this, because when the molecule attaches to the ion channels, they actually make the open close motions. And then once it's opened, so current can measures. Although the size of the amplitude of the current is only 10 picoampere and so on, but we can actually see the difference between these states. Okay, and also the selectivity, as I mentioned, is very nice. This is a kind of human sweat odor, and the molecular structure is like this. And if we use the very similar molecules or molecular weight, and we can actually recognize the difference between these kind of molecules. So the selectivity is really high. Okay, so this is a very simple demonstrations. And we actually absorb the human sweat to the paper like this and approach to the robots, and the robot actually escape. So running out from the human odors. That is that's not the goal of our research, because the robot has to find the human beings in the near future, so that we need to change the algorithm to find the source of the odors in the near future. Okay, so this is a kind of the results of example of detection of the human odors. But if we change the type of the membrane proteins, so we can actually detect the other type of the molecules, like molecules from the addictive drugs or bombs and so on. And also that's kind of the sensors can work for the food, cosmetic, agriculture, or environmental monitoring and so on. But I believe the huge market area is the healthcare applications. So if we can actually detect the molecules coming from the mouth, I mean the breast, so that we can actually detect the cancers or diabetes at the early stage without the invasive way and so on. So that we first try to detect some molecules coming from the breast by using our biohybrid sensors. So about 2 years ago, so we first had the very nice result to detect the cancers coming from by using the mosquitoes sensors. So this is actually experimental setup to detect the breath. And here is the kind of the gas bag collecting the breath of the patients. And the gas go through the channels and to the sensors. And this is a kind of sensor box having the multiple sensors, but all the sensors are all multiple sensors, but all the sensors has the mosquito sensors that can detect the cancer molecules. Okay. Okay, so this is a real experiment how it's going on. And after actually, so this is one of our researcher in our group. He is very healthy, not the cancer patients, so that his breath contains any of the I'll say none of the cancer molecules. So that after correcting his breath, we actually put the cancer marker to his breath. very small amount of the cancer marker to his breast, okay? Like this. And without any, how to say, pre-processing, so we directly put the human breast onto our sensors without any filtering or pre-experiment and so on. But so we can actually detect some signals coming from our sensors. Oh. Okay, just a moment. So the sensor signals coming out like this. So those open-close state is very important to detect single specific molecules. So this is actually experimental results. So without having the, how to say, cancer molecule, cancer markers, so we don't have the open-close signals, okay? But once we put a very small amount of the cancer marker, which is about the 0.5 ppb, so we can actually see the open-close state like this. So this concentration is very, very small. So thinking about the gas chromatography, you can actually detect the gas from the breast, but they cannot detect the 0.5 ppb without some extractions and so on. So even if you use a desktop and very expensive machines, we cannot detect the 0.5 ppb. And of course, if you increase the concentration from 10 times, like a 5 ppb, signals becomes much more obvious and much more clear, and gets the increased open-close probability from the single molecules. So those kind of the, how to say, signals might be very useful for detection of the cancers or even diabetes and so on. So that's biohybrid sensors might be very useful for very small, but the highly sensitive and selective sensing in the other futures. All right, for the sake of time, so I would like to summarize my talk. And I actually introduced of the biohybrid robotics that can be divided into 4 groups. One is the biohybrid sensors, reactors, and processors and actuators. But with those kind of, by the combination of those kind of devices, so we in near future want to combine everything in the single systems and make a humanoid robot in near futures. But these kind of the work itself can be applied to a lot of the research field or industrial fields like a sensor industry or medical, pharmaceutical, or robotics industry as well, and even the food industry. So that those kind of the how to say research like a biohybrid robotics might be very useful for the various type of the research and industry. With that, I would like to thank all the group members and their financial support. Thank you very much for your attention.
Okay, thank you very much, Shoji. This was a very, very impressive talk introducing all the different technologies of biohybrid robotics. And we actually got quite a few questions in chat or also in Discord. So maybe you cannot ask all the questions here, but maybe Shoji can answer to these questions after the session. One of the questions we got here, a very interesting one, is how to control and keep this living muscles or other cells. How can you stimulate them? Is it easy? Is it difficult? Can you just tell us a little bit more about how to deal with these living cells?
Okay, so again, so I can show you the movie how it works. So we can actually actuate the fingerbone-like structure like this by the muscle contractions. And we can actually selectively control the muscle contractions by applying the electric field selectively. So those kind of the also, okay, I can show you this slide much better to understand. So these 2 electrodes that can only make the electric field is this how to say cut these regions, so that once we apply the voltage between these electrodes, the muscle contract only in these directions. And when we apply the voltage between these directions, these electrodes, the muscle make a contraction until this direction, so that we can selectively apply the voltage between the muscles. So, and also we did this experiment in the solutions. Solution is like how say KCl or PBS and so on. So that is a kind of the conductive solutions, so that we can just put the electrodes here to here and make an electric field between the muscles. That's why the muscle can actuate selectively. Okay.
Okay, thank you very much. I actually have a question to you from myself, and especially a lot of embodied intelligence researchers in this community interested in how the information processing, computation, and all of this, the information processing aspects can come out from physical embodied systems. And I think one of the slides you showed about how to do biohybrid for computation. Can you actually give us a bit of comments about what's your views about the information processing, how the information processing actually happens in biohybrid systems?
Yeah, that's a kind of the future work. We are now planning to do so, but we actually culture the living neurons at a single cell level and make an artificial network, artificial network by using the living neurons and so on. So if you can make such kind of neural network, it can be work as a physical reservoir. So that's if you can make the control of the neural dynamics, and we can actually calculate something, so learn something by using this kind of network, I believe. But that is a kind of the future goal of our research.
Do you think this is going to be any better or any different from a conventional silicon-based computational processes? What's your views on how
perspective?
different, how better, or how yeah, you think?
I'm not sure that silicon processor has the very high and can make some multiple processing at the same time. But I think the living cell, one of the advantages of the living cells is the energy efficient, so that we don't need so much electricity or carbon footprint and so on. That might be one advantage. And also I believe the multiple calculation is possible, or the highly parallel calculation is possible compared to the silicon processes. But I cannot say exactly at this moment.
Yeah, excellent. Yeah, thank you very much. And there are lots of questions related to your biological skins around this robot fingers. And one of the questions was about how the stiffness, the modulus of the skins. Is it going to be as soft as the human skins, or is there going to be any limitation in Young's modulus and stuff like that?
Yeah, we actually didn't to measure the exact stiffness of the skin, but let's say if you see the movie like this, so it looks like a real epidermal layer of the skin, so that we believe the stiffness itself is very similar to the one of our human skin. But again, so this is only a structure of the two layer of the skin, but the actual skin has a lot of the other other functions or other components, like the component of the sweat, or component of the hairs, or component of the some oils coming out from the skin and so on. So those kind of structures might be very important to mimic the real humans in the near future.
Right, and actually this leads to the next questions about the integrations of this mechanism. You have a very impressive technologies in component levels and muscle sensors, skins, and so on. What's your views on how to integrate them together? Or because I think this is one of the biggest challenge of how to handle biological cells, and if we put these two mechanisms together, is not necessarily working as we expect in individual and like in individual function. What do you think about the future of integrating the different mechanisms in biology?
Yeah, that's a good question. So one of our, well, I think the most of the students in my group want to combine the many components into a single one. And for example, so they actually want to combine the muscle tissue with the neural fibers, neural tissue. So that's the in the near future, we can actually make the muscle or control the muscle contractions by the signals coming from the neurons. It looks like our human beings. And also, we actually would like to also actuate this kind of finger bone-like structure by using the power of the, I would say, muscles in the other future. So that the combination of those kind of the, I would say, tissue, like muscles, blood vessels, neurons, and skin is our next goal. And to do so, we actually have a lot of issue. For example, if you want to make a very thick tissues, we need to perfuse the nutrition into the deep area. Otherwise, the tissue may damage very quickly. So that's a cultivation of the also elongation of the lifetime of the tissue is also a very important issue.
Excellent. Yeah, thank you very much, Shoji. I think there's still lots of questions in the chat and the Discord. So if possible, I hope you can join these questions and answer to these questions individually. And also, if possible, you can stay for another an hour or so that you can join this panel discussions in the next session. So. In any case, I think we're going to stop here for the first panel talk by Shoji. And thank you very much once again for a wonderful introductory plenary talk for this Embodied Intelligence Conference this year. So we'll have a 15-minute break, and then we're going to have the first short talks and panel sessions in 15 minutes time. So at 10:30 in GMT. Thank you very much, Shoji. See you around. Thank you.
Yeah, thank you.